Hello everyone, welcome back to class. In daily life we all know that oil and water do not mix. The hydrophobic region of the cell membranes, lipid bilayer, forms a natural barrier, preventing most substances from crossing freely. So the composition of solutes inside and outside the cell differ markedly. However, during cellular metabolism, the cell must take in oxygen and nutrients, and also expel metabolic waste products. The movement of these substances occurs across the cell membrane. So how do substances cross the cell membrane to be transported? First, we classify modes of substance transport. We categorize them according to size and energy use. By the size of transported substances, there are two main categories. Small molecule transport, which moves water-soluble small molecules, such as carbon dioxide, oxygen, amino acids, glucose, and charged ions. Among these, based on energy consumption, they are divided into active and passive transport. Large molecule transport is mainly for transporting large molecules and particles, such as proteins, bacterial fragments, and hormone secretion. We will explain each type in turn. The first mode is simple diffusion. Simple diffusion is a physical process. According to the principle, like dissolves like, highly lipophilic substances more easily cross the bilayer. Therefore, substances moved by simple diffusion are mostly lipid-soluble or some uncharged polar small molecules. For example, oxygen, carbon dioxide, nitrogen, steroid hormones, ethanol, urea, glycerol, and water. Characteristics of simple diffusion: no energy consumption, moves down concentration gradients, and does not require protein carriers. Its rate mainly depends on the concentration difference across the membrane and membrane permeability. Next is facilitated diffusion, also a passive transport. Facilitated diffusion refers to transport aided by membrane proteins, in which non-lipophilic small molecules or charged ions move down concentration or electrical gradients across the membrane. Depending on the transport protein involved, facilitated diffusion is divided into two forms: channel-mediated facilitated diffusion and carrier-mediated facilitated diffusion. Channel-mediated facilitated diffusion occurs through channel proteins. It mainly transports various charged ions and is therefore called an ion channel. Ion channels have two important features: selectivity and gating. Ion selectivity means each channel is highly permeable to one or a few ion types, while being poorly permeable or impermeable to others. For example, the N2-type acetylcholine receptor on the postsynaptic membrane of the neuromuscular junction allows Na+, K+, and Ca2+ to pass, but predominantly conducts Na+. Gating is a descriptive term. Much like the act of opening a door at home, most channel proteins contain movable structures that act as gates. Various factors can trigger gate movement, opening or closing the channel, a process called gating. According to the gating stimulus, channels can be classified as voltage-gated channels regulated by membrane potential, such as voltage-gated calcium channels at nerve terminals, chemical-gated channels regulated by ligands, for example the N2-type acetylcholine receptor, and mechanosensitive channels controlled by mechanical stimuli, such as mechanosensitive K+ channels in cochlear hair cells. These gated channels are like locks opened with keys, such as fingerprint or facial recognition locks. Carrier-mediated facilitated diffusion refers to water-soluble small molecules or ions being transported across the membrane down their concentration gradient, which is a carrier-mediated passive transport. Many important substances in the body are transported across membranes via carriers, for example glucose and amino acids. Carrier-mediated facilitated diffusion has these features. First, structural specificity. Carriers recognize and bind only specific structures. Second, competitive inhibition. If two similar molecules bind the same carrier, they compete with each other. The transport of the lower concentration substance is more easily inhibited. Third, Saturation kinetics, because carriers and their transport rates are limited in the membrane. When substrate concentration increases beyond a point, the transport rate no longer rises with concentration, which is called carrier saturation. If we compare the phospholipid bilayer to a small river, carrier-mediated diffusion is like using a small boat to carry goods between the banks, whereas channel-mediated diffusion is like building a bridge across the river to transport goods. But this bridge has gates, which open only under certain conditions. Simple diffusion is like swimming across directly. These transport modes do not consume energy, so they can only move substances down concentration gradients, that is, from the high to the low concentration side. Besides passive transport that does not consume energy, some cellular transport processes require energy expenditure. We call energy-requiring transport active transport, depending on whether the transport protein directly uses energy. Active transport is classified as primary or secondary. The term direct is key here. What does direct mean and how does it differ from indirect? Let's look further. Primary active transport means the cell directly uses metabolic energy to move substances against their concentration gradients. These transported substances are usually charged ions, and the membrane proteins mediating them are called ion pumps. For example, the Na plus K plus pump moves Na plus and K plus simultaneously. The Ca2 plus pump transports Ca2 plus, and the proton pump transports H plus. These proteins can directly use ADP. Note the difference between pumps and channels. Although sodium channels and sodium pumps, or calcium channels and pumps, transport the same ion types, ion channels do not consume energy and only allow ions to move down their concentration gradients. Ion pumps consume energy and can move ions against concentration gradients. Some active transport processes are driven not directly by ADP hydrolysis, but by the ionic gradients established by primary active transport, using that electrochemical potential to drive transport against gradients. This indirect use of energy is called secondary active transport or cotransport. Because two substances are transported together, it can be classified as symport or antiport based on direction. When transported molecules move in the same direction, it is called symport, and the carrier is a symporter. For example, glucose absorption in the intestinal mucosa and reabsorption in the proximal renal tubule are achieved via the Na plus glucose symporter. Amino acid absorption in the intestine also occurs via Na plus amino acid symporters. Additionally, Na plus K plus -2 Cl symport in renal tubular epithelium and Na plus I symport in thyroid epithelium are examples of secondary active transport. When transported substances move in opposite directions, it is called antiport or exchange, and the carrier is an exchanger. Two important exchangers in humans and higher animals are the Na plus Ca2 plus exchanger and the Na plus H plus exchanger. In most cases, the driving force for secondary active transport comes from the transmembrane Na plus gradient established by the Na plus pump. In diabetes rehabilitation, appropriate exercise therapy can increase the number of glucose transporters on tissue cell membranes, promoting glucose transport and utilization by tissues. The body contains not only water-soluble small molecules, but also many insoluble large macromolecules. These substances cannot cross the cell membrane directly, but are enveloped by membrane to form vesicles, and transported by processes of engulfment, membrane fusion, and membrane fission. This series of processes is called vesicular transport. Vesicular transport is an active process requiring energy and numerous proteins, and involving changes in cell membrane area. Vesicular transport includes exocytosis and endocytosis. Exocytosis refers to the process where large molecules within the cytoplasm are expelled from the cell in secretory vesicles. For example, exocrine gland cells release zymogen granules and mucus, endocrine gland cells secrete hormones, and nerve terminals release neurotransmitters. all of which are exocytotic processes. During exocytosis, under the mediation of many proteins, vesicles move toward the inner surface of the cell membrane and fuse with and rupture the membrane, ultimately releasing their contents outside the cell. Endocytosis refers to extracellular large molecules or particles, such as bacteria, dead cells, and cell debris, being enclosed by the plasma membrane and entering the cell as vesicles, also called internalization. In contrast to exocytosis, endocytosis reduces the cell membrane area. It is worth mentioning that, whether exocytosis or endocytosis, both are more complex than active and passive transport.